This mini lecture is going to talk about the other parts of Chapter 12. So Chapter 12 is about the cell cycle, and it's about that biochemistry of regulating the start and the G2 to M transition. There's also a mitotic checkpoint. Those are big regulatory steps in the cell cycle, and we really focused on that with a big handout. Describing the activation of CDK1. There are other CDKs that are active over here that are the targets of what? Of the PDGF signaling pathway. So PDGF acts to regulate skin proliferation, skin cell cycle, and wound healing by regulating CDKs at this point of the cell cycle at start. So remember that we zoomed over here to look at this checkpoint in order to understand this start control point a little bit better. Okay, so that's where we're at, and we are now going to talk about the other piece of Chapter 12, and the other piece of Chapter 12 talks about interphase and then the steps in mitosis. And that's another piece, and we're just going to cover those now. So interphase occurs and includes G1, S, and G2. And so let's just look at a little summary figure. Interphase includes G1, S, and G2 of the cell cycle in eukaryotes like humans and other eukaryotes, and then mitosis is M phase. Interphase includes cell growth. It includes that start point of the cell cycle. It includes DNA replication. And it includes something that scientists refer to as G0. And G0 is often drawn like this, where there's sort of a mini side pathway to the cell cycle, and it would occur here at start. And it's a little bit inaccurate because really the cells are just waiting at start like a car at a red light. And they don't do anything too special, but there are several examples, many examples of cells that spend a long time in G0. For example, the cells in your brain that are no longer going to divide again are in G0. When we were kids and we were growing as boys and girls, there was a subset of our cells that were in G0 waiting for the hormones of puberty to stimulate secondary sex characteristic growth and maturation of men and women. So those cells, when we were young, were waiting. And that's an interesting time because as a child, some parts of our body were going rapidly and others weren't. Others were waiting in G04. They were waiting at start for hormones to stimulate cell division. And there are many examples of those. And so scientists often refer to this G0 phase of the cell cycle, which is really when the car is just parked, sometimes for a really long time, at a red light. So G0 occurs in interphase. And then the next couple of figures show what happens in mitosis. Okay, so in the cell cycle, we have interphase, and then mitosis, interphase, and then mitosis. These important things happen during interphase. What happens during mitosis? And we're going to summarize what is too much information in the book. And here it is. We're going to talk about prophase within mitosis. We're going to talk about the steps within mitosis being prophase, metaphase, anaphase, telophase, and cytokinesis. And we need to learn those terms, and we need to learn one thing that happens during those phases. And prophase, the chromosomes become visible. Because they condense. The chromosomes are very long, and they need to be packed in order to fit inside the nucleus. And but in interphase, they're packed loosely, and they're not visible under a microscope. At the start of mitosis, it's indicated under a microscope because the chromosomes become visible. The chromosomes further shorten or condense. And that's the result of CDK activity. CDK is a kinase that phosphorylates. Here's some DNA, and the DNA is wrapped around proteins called histones, and they are one target of the CDK1 kinase. When phosphorylated, the histones pack more tightly together, the chromosomes shorten, and they condense, and they become visible. So what happens during prophase? The chromosomes shorten, become visible due to condensation due to the activity of CDK1 kinase. Then metaphase, the chromosomes line up in the middle of the cell. Why are they lining up? They're attached to parts of the cytoskeleton. They're attached to microtubules. So we should learn that. Remember, we learned some about what the cytoskeleton components are, and one of the things that microtubules do is they move stuff around inside the cell, like here, when they're moving the chromosomes. Now the chromosomes separate, and they move to the opposite ends of the cell in anaphase. So chromosomes separate. And if you could learn one thing that in this one step, this is the most important step in anaphase. Then in telophase, the nucleus starts to reform, and cytokinesis is cell division. And the nucleus starts to reform because of the polymerization of intermediate filaments, another component of the cell cycle. And this cell starts to divide because of contraction of actin microfilaments. There are lots more details shown in the figures and in the text, but if you could learn one step from each of these phases, prometaphase is just a kind of transitional phase. We don't need to worry about that. Chromosomes line up, chromosomes separate, and the nucleus reforms and the cell divides. That would be awesome. The book goes into great detail talking about the centrioles copying, dividing, and it's possible that some additional questions will come from the test. So you're in the test, so you're assigned to read through this material, and but focus on the highlights. And that's that. This is just showing in detail, zooming in, those steps: metaphase, anaphase, telophase, cytokinesis, like that. And that's the end of chapter 12. Just to go back to where we started in the beginning, this shows cell division in eukaryotes, and I mentioned this in an earlier video. In prokaryotes, cell division is called fission, and it's different than eukaryotes. In eukaryotes, though, like we've been talking all of chapter 12, all the same ortholog proteins like CDC25 are found in every single eukaryote. It's really an amazing story. And that's the end of chapter 12.